Okay. Good afternoon. My name is Doctoral Aguilar-Rabiela, and I will talk a little bit about my project, Microspheres for Novel, Localized, and Specialized Treatments. My advisor is Dr. Berenice Vergara. Okay, have you ever noticed that the conventional methods for drug administration have not changed since about 2 centuries? This represents some issues in terms of the typical pharmacokinetic curve, in which we have a minimum efficiency concentration and a minimum toxic concentration, and we just have a limited window of time for the effectiveness of the drug. Of course, this represents some problems in terms of the efficiency, the drug stability, the side effects, if we are working near to the toxic concentration, and the limited administration pathways. Mainly we have oral and through injections. So the proposed solution is the use of microspheres as drug delivery systems. But first of all, we have to talk about what a microsphere is. And contrary to the concept of the capsule, in which the drug is inside a membrane or inside a shell, and when this shell breaks, the whole concentration of the drug spreads away, the microsphere is more similar to a cookie, in which the drug is dispersed through the whole device. In this case, the whole dispersed through the polymer matrix. So some important aspect during the fabrication of this kind of devices is the size. Usually, if we are working between 1 and 100 micrometers, it's considered microscale. If we are working below 1 micrometer, it's considered nanoscale. And it will impact the interaction between the device and the biological environment. Another desirable properties for this kind of devices is biocompatibility and biodegradability. That's why we choose a biopolymer called polyhydroxyalkanoate, or PHA, for the development of our device. What are the advantages of using microsphere for controlled release? For drug delivery, is that we can allow controlled release. The mechanism of transference of the drug from the microsphere to the environment could happen mainly by 2 phenomena, diffusion and degradation. Due to the modification of the properties of the device, we can control how the drug will release to the environment. So we can reduce the side effects to having high concentrations in the environment. And of course, we can explore new administration pathways. As I mentioned before, the size of the particle will impact in the interaction with the body. So if the device is small enough to cross or to penetrate some membranes or tissues, the device can easily reach the veins, the arteries, and even the blood vessels. So we can administrate it through intraarterial and even by inhalation or through eye route. So we can explore new treatment ways. So the current result is that we have developed microsphere of PHA for controlled release of curcumin. We are working on the microscale, about 6.9 micrometers, with an entrapment efficiency of 98%. We also have tested this device on simulated body fluid for the obtention of the release kinetic. And this release kinetic has been mathematically modeled. The results of this work actually has been published in the International Journal of Biological Macromolecules. And you can find it under this name. Then we test these microspheres on cell culture by using the cell line I549. And we test the cell viability through 2 different methodologies, MTT and flow cytometry, and at 2 different time sets, 6 hours and 24 hours. During the first 6 hours, we observed a strong difference between the administration of the same amount of curcumin through conventional methods compared to the administration through this microsphere. So we can talk about a controlled release of curcumin in terms of the delay of the apoptotic effect observed. And the result was confirmed through the 2 different methodologies used. Then we also worked in collaboration with the Biomaterials department at the Friedrich Alexander Universität, in which we explored new approaches about the use of microspheres like this, such as new methods for drug delivery tracking through the incorporation of biomarkers inside our microspheres, and the development of 3D models for the study of the interaction between the cells and the microspheres. And more interesting, we also incorporated nanoparticles of some novel materials such as pions, which are paramagnetic particles into our microspheres in order to move it or direct it to specific zones or specific tissues. And with this, allow target delivery, and maybe in the future, new localized cancer treatments. And finally, we incorporate also bioactive ceramics, such as nanobioglass, which has exhibited bioactivity in terms of the formation of a layer of hydroxyapatite on its surface. This material is very important for the bond formation. So we can use these composite microspheres in bone tissue engineering application for the development of new osteoporosis treatments. In summary, the importance of the use of microspheres for these applications is that we can allow control release, and through the incorporation of novel nanoparticles such as pions or nanobioglass, we can develop in the future new treatments, new localized cancer treatments, or even new osteoporosis treatment. And of course, continue with the study of the interaction between microspheres and cells through the incorporation of biomarkers on it. Thanks for your attention.
Muchísimas gracias por